organization. Also some insights about superficial white matter tractography, some atlases that have been created for its analysis, a few examples of the analysis of superficial white matter in disease, and some new methods for the analysis of these fibers. So please, if you have any question or comment, just let me know. So we have for the white matter different kinds of categories. So we have the projection fibers connecting the cortex with other structures, commissural fibers connecting both hemispheres, and association fibers connecting regions from a same hemisphere. And between the association fibers, we can separate them into long association fibers. Sometimes they are also separated in medium range fibers and short association fibers. And between the short association fibers, we have the U fibers that are follow the morphology of the sulci and are very short. And we have in the superficial white matter that is the layer of white matter that is close to the cortex, under the cortical layer. We have fibers that are parallel to the cortex, and other fibers that are radial to the cortex. So we have these two kinds of fibers, and we will see that with tractography we normally have an easier tracking of the fibers that are parallel to the shape of the cortex. And the other thing is the fibers are organized depending on their length. So the shorter fibers are closer to the cortex, and then the longest fiber are deeper in the white matter. So there are several things about these connections. First, cortical fibers, the short connections, so connect regions of the same gyri or the neighboring gyri. They are superficially located, as I said, and their number is inversely proportional to their length. So here in this picture, we have the very short fiber. These are intracortical fibers, not superficial white fibers, are the more numerous fibers. And then we have the U fibers that I have to say that there are a different classification of U fibers. For example, here in the work of Tuesday are classified between 3 and 30 millimeters. Other works use up to 40 millimeters. In some tractography works, these are classified up to, I don't know, 50 or even more millimeters. And then we have the long fibers between 30 and 100 millimeters that are inferior in number. So the important thing is that short association fibers are much numerous than the longer fibers. Okay. And the other thing that is important to highlight is that superficial white matter is a different tissue layer than the deep white matter, and it is composed of different kind of structure axons, but also subcortical neurons, glial cells, and vasculature. And this, I think, is very important because it's a different layer than the deep white matter. So this layer has different rate of maturation, and some regions, for example, the frontal lobe, takes more time to mature, and also the myelination process is late than in the deep white matter, and it can take up to 30 or 40 years old. And this may this may produce a more vulnerability of this white matter to myelin-related disorders. And the other important things is that white matter, superficial white matter, has a big amount of interstitial neurons, and their function varies across the superficial white matter. So it seems that there are also, as in the cortex, different functions and regions between the superficial white matter. And these are located in all under the cortex in all the brain, and their density but also varies across the different regions. So this picture, I really like it, from Van Dikens and colleagues' work. It's a very recent review, and I think it summarizes very well what we know about superficial white matter. So here we can see the two kinds of fibers, the transverse fibers under the cortex and the radial fibers of the superficial white matter. And here we can see, can you see my pointer? I'm sorry, I did not ask. Or not? Yes?
Yeah, I can see your mouse.
Yeah, okay. So here we can see that in diffusion MRI, we can better track the transverse axons. So that's what is related with the chiral bias, that where most of the fibers are tracked up to the crown of the gyri, and it's more difficult to track the fibers that are radial to the whole gyri. Also, there is a difference in the vasculature in superficial white matter, and what is very interesting is the large amount of neurons, interstitial neurons, in this tissue, that also is a way to differentiate this layer from the diffusion, from the deep white matter. And also the iron concentration of superficial white matter is different, and the myelin also has a gradient in this region. So this can make possible to distinguish this layer using MRI. Okay, so how can we study superficial white matter bundles? We can use dissection, the Klinger's method, which enables the identification of some fibers, but is difficult to apply, and is not 100%, I will not say reliable, but in sometimes it's difficult to extract some fibers. So for example, here is the vertical occipital fasciculus that can be seen. So what is interesting anyway, is with this kind of method, is possible to validate the finding with diffusion MRI tractography. And I'm very surprised because in this work you can search it from Van Dieken, and they list a very large number of superficial white matter bundles that have been already validated with this kind of dissection. So I was very impressed about that. And this is a very new work, very interesting, also from this year from the Nothoff, where they use the same kind of Klinger's method, maybe with some variation. But the important thing is that the strategy of peeling the brain and the white matter is from inside to outside, from the internal part of the brain to the cortex. And this enables the dissection and the obtention of the interchiral fiber layer in the cortex, and this is very beautiful, I think, because we can see all this layer. This is the lateral aspect of the right cerebral hemisphere, and also we can see the medial aspect. We can see the different layers. So this work, I think, and this method that's very new, I think, is very interesting. So the Klinger's method, I will not detail it, but it's very complex and has several steps to get the water crystallized in the white matter tissue. It's very interesting. So another thing that was very interesting in this work is that we can see very well the superficial white matter layer. Here you can see, and its correspondence with MRI, T2, ex vivo T2 MRI. And we can see that the thickness of this layer is about, is maybe up to 2 millimeters. And this is an important thing that is discussed in the paper, that with diffusion MRI, it seems we overestimate the thickness of this layer. And another important thing that we can see here is that the thickness varies across the cell site. You can see that even in one cell site is different in the different regions of it. Okay, this was the more I will see morphological part. And now I'll talk you a little about diffusion tractography and the study of these fibers using different methods. So I would say that some very I would say seminal works were performed by Catani and other researchers also, that try to segment these bundles using manual segmentation, mainly based on the manual manual or semi-automatic placement of ROIs in the cortex. So this finding was very interesting because they show that it was possible to segment specific white matter, superficial white matter bundles. And also another work from Braun shown that using the connections of these superficial white matter fibers and the density, the connections is possible to distinguish a particular connection. And this is very interesting, I think, this kind of strategy, because it's related with what histology says, that the density is different in this in these connections. So I think it's something important to take into account for the analysis. So what is difficult in for the analysis of these fibers is that they are very related to the morphology of the subsite, and we know that this morphology is very different between subjects. So the methods, it's difficult to develop methods that can identify the same values across subjects. And this strategy based on, for example, density and along the subsite may be a good approach to achieve a good segmentation, but it's difficult to have this kind of method working in the whole brain. So that's why the other strategy, more exploratory, is to create superficial white matter bundle atlases for the whole brain using several subjects and base it in general on fiber clustering to get the mean, I would say the mean or the most stable connections across subjects. So there have been several atlases created, and we can see that they have increased the quality along with the quality of diffusion MRI acquisitions and diffusion models and tractography methods, and other also post-processing algorithms such as filtering or mainly filtering, I would say. So, for example, here I will show these two atlases were created with the same database, a Harvey database, but the difference is that the second atlas was created with based on nonlinear registration. So we can see that the registration, of course, is very important for this kind of fibers. And also the last one here was created with a more specific registration that also takes into account, of course, the T1 image, but also the segmentation of the subsite. So this method could get a better segmentation and characterization of the superficial white matter, but it's more difficult to apply, of course. It requires a better registration. And also there have been developed new atlases based on the HCP database and with more bundles, up to 4,000 or 400 or 500 bundles. So these atlases represent the mean shape of the connections, but they are still not very related, or the relation with the cortex morphology is still not far from being perfect. But in any case, these atlases have enabled the study of these bundles, and also this is just an example of some connections that have not been studied at the population level, for example, the insula connections and other kinds of connections. So these are useful for this kind of general description of the bundles. So this is just an example. I apologize, as I said, because I have, of course, more knowledge about the works that we have, but there are many other works interesting work from other groups. So it's just an example for people who have not seen yet this kind of superficial white matter bundles. So they cover the superficial white matter where the long fibers have not complete coverage of this region. So it's interesting to have this, consider this analysis for studying different populations. Okay, so when we have an atlas or another method, we can identify the fibers in other subjects to perform the population analysis. And this is just one example, but based on the atlases, we can segment the bundles in the subject. Here, I had an example, it's a very low quality database, clinical database from Chile, very bad, but just an example of the segmentation of this bundle. And you can see that for this very low quality, the segmentation is not precise, but anyway, it gives some localized information about the superficial white matter. It can be improved, of course. It's just a raw segmentation. And there are many analyses that can be performed, but the simplest one is to, from the segmented bundles, we can calculate mean diffusion-based measures such as the fractional isotropy or mean diffusivity, etc. It's more difficult to relate, to study, for example, or compare the number of fibers. Maybe the bundle volume is more feasible. And just two examples for a study of autism spectrum disorder based on the superficial white matter. This work could find a difference in the generalized fractional anisotropy with a decrease in this measure related to several clinical scores associated with empathy and social abilities. So what is common in this kind of bundles is that the findings are spread across all the brain, I would say, are less for different disease is the same thing. I would say the change don't present a pattern or very localized differences. This is very spread along different parts of the brain. And also, this is another study with patients with early multiple sclerosis. This was a very small sample, and this is a problem with clinical data. But in any case, we could find some alterations in superficial white matter, and also some of these related with clinical scores. So here, I'm not showing here that, but what was interesting is when applying machine learning method, the more important features were related to superficial white matter, more than cortical features and than white matter features. So in this case, it seemed that superficial white matter were presented more alterations in the early stage of multiple sclerosis. So what I will show here is just another a few methods that try to improve the segmentation of these bundles and the study of these bundles. So as I mentioned before, we have the Jaral bias that tends to track the streamlines parallel to the circumvolution, and all the fibers connect the ground of the gyri. But we know that fibers are radial and connect all the gyri. So with standard tractography, we miss this other connection, and sorry, it's here. There has been developed some method to improve the tracking. This is one, but there are others, also new works that try to track this to perform a better tracking of these kind of fibers using, in general, the cortical surface, not the cortical, the interface between gray matter and white matter surface, and using the direction of this mesh, they can guide the tractography and have a better reconstruction of the streamlines. So I think these are very interesting approach to improve the quality of the tractography for these fibers. Also, there are other methods that try to filter the superficial white matter bundles. And here's just an example using convex Hull and identify a main fascicle of the superficial white matter bundles, would enable a better retest reproducibility and also more significant difference between populations. So that shows that filtering or a specific method for superficial white matter tractography are needed to obtain a better quality of or a better significance, we can see, of the analysis. And I missed this one. This is another example. Also, we can improve the segmentation methods themselves. For example, this one is a deep learning-based method that use a classification of superficial white matter and non-superficial white matter fibers that help to better identify which fibers are from the superficial white matter and a better labeling of these fibers in new subjects. So this is another strategy to segment these fibers, and this work shown that it works very well, even with, for example, brains with tumors. So I think this works very well for medium range fibers. At least for medium range fibers, it works very, very well. And I think this, just one last example. This work is from Chile, and it's very interesting because it shows the advantages of the diffusion-based analysis, in this case for superficial white matter, because using these automatics and specific methods, this work could analyze a very large database along a big lifespan. And here we can see the segmentation that was performed of the superficial white matter, and several analyses could be performed for this population, for example, to characterize the thickness across different brain regions and different age. And also an interesting finding was that the superficial white matter volume changed, but as the other tissues, but the trajectory is slightly different. But it was most relevant is that the relative fraction of the total volume of superficial white matter increased with age. And this was the opposite for the other tissues. So this was very interesting. And also this work could characterize and associate the thickness with other features of the cortical thickness and curvature. So I think that is. So thank you very much.
Thank you very much, Pamela. Can you hear me?
Yes.
Thank you very much for your presentation. Very interesting. I'm sorry I missed part of it because everything crashed on my side, but I came back to the lab upstairs and I cut most of it. So we would open the floor to question. If there is anyone with a question, you can ask in the chat or you can unmute yourself and ask. I would like to start by asking something about superficial white matter atlas and segmentation. Every time I see an atlas or paper that says we segmented 100 superficial white matter bundle, it seems most of them are in the frontal lobe. And it seems like the frontal lobe is either the easiest to reconstruct or the one with the most focus. And sometimes the temporal lobe has almost no bundle, or it seems to be more difficult to track superficial white matter in the temporal lobe. And there is nothing in the insula. Is it the case of easiest to track and hardest to track, or is it just a matter of number of gyri and then like you have more combination in the frontal lobe?
It's a good question. Yes, that's true. That I just go to the. Yeah, it seems, for example, you can see here the temporal lobe, there are less fibers here, no, in the atlases. So I think it's more difficult. Maybe there is more variability. In the case of the frontal lobe, there are very stable and similar bundles across subjects. So that's the reason, because it's bigger, because I don't know, there are also very stable areas, for example, in the precentral postcentral connections that we can get easily. So for temporal lobe, yes, it's more difficult. And other areas also in the medial region of the brain that are more variable, and the fibers, the shape of the fiber is different. Not very U-shaped, so it's more difficult.
Perfect. That answered my question. Thank you. Any other question from the crowd or online? If you ask in the chat, we can read it.
Well, there was a question, Pamela, in the chat regarding the references. So you provided a new list of recent papers. So I told Diane that we would email them your list of references after your presentation.
Yes, of course. I confess that in this presentation, I have to update the reference, but of course I update the reference and send you the presentation. Yes.
Great. Maybe a quick question from me. Do you feel we need tractography specific parameters and methods just for superficial white matter, given their particular anatomy and also this concept of layers, and maybe code the rules of deepness, things that are closer to cortex and deeper? Do you think there's a need for a new generation of dedicated algorithms just for that?
Yes, I think, and also I think that what a problem we find is that for the analysis of clinical database, commonly we don't have enough subjects. So if we have an atlas of, for example, 500 bundles or more, when we try to find significant difference, it's not possible. So I don't know. I think maybe we should focus on identify very stable bundles and develop methods to very well extract these specific bundles related to the morphology of the cortex, where we have higher density of the fibers, etc. So to maybe to identify less superficial white matter but with more reliable and specific methods. And I think this will bring a more better result for the population analysis.
I would be curious to hear, Alessandro, people about global approaches or commit-like approaches on such structures where clearly we see that maybe the myelin content is different, maybe the diffusion signal is quite particular in these layers. I don't want to put you on the spot, Ale, or anybody, but I'd be curious to hear your thoughts on commit-like strategies or global approaches here.
I can try to comment, but you can hear my voice. It's very
good. It's good. We can hear you.
No, I'm curious about these atlases because indeed I was thinking while Pamela was speaking that we could use these atlases as a combination with tractography and then to try to see whether they survive the filtering or not. Because perhaps we are not able with tractography to recover them, but perhaps injecting these priors into the tractogram, we might be able to find them, not directly, but indirectly. So I have a question for Pamela. Which of these atlases are open to your knowledge?
I think at least these 4 atlases are open. This last one I don't know, but the other yes, they are open. But yes, I think I'm performing this review. Now I'm convinced that superficial white matter has a very, it's like the cortex, very different features across the cortex. So I think we should use this information, cortical information, I don't know, functional information, etc., to identify these bundles, not only the tractography.
I see.
Anybody else from the room? Paolo, I see you open your camera. You have a question?
Yeah, I have a question. Thanks of all. Okay, thanks, Pamela, for the overview of this very interesting topic. I have the assumption that, of course, registration play a key role in the building of Atlas. And I have this bias that as much you move to the border of the brain, registration became harder and harder. And so when you have to build an atlas for deep white matter, so the main bundle, okay, you can expect that the variance is quite small. But as much as you move to the external part, so the U fiber, it became the variance became larger, and then build an atlas of surface white matter, superficial white matter might be harder. Did you have this evidence, empirical evidence, that the variance of reconcile all the cluster from different subject is harder than just deep white matter?
Yes, it's difficult. Even the tractography itself is more difficult in superficial white matter. We need, for example, use probabilistic tractography and maybe, as Maxime told, specific parameters. And then for it's very their identification is very dependent on the morphology and the registration. That's clear. So, and I think we need for the segmentation, we need a good registration and also identification. Maybe not separate identification of each bundle, but identification of patterns of bundles to try to identify the different morphology of the superficial white matter that change in the subjects, but change a little bit in subject. I don't know if I express myself really well, but to use the information from regions, not only separated bundles for the segmentation.
Question from the room. There was a raised hand from Kurt and Lauren. I don't know who.
Kurt is first.
Kurt?
Yeah.
Kurt, can you go ahead?
Hi. This is actually a question that came because of a meeting that's run by Lauren tomorrow, the International Society of Tractography Definitions Unit. And I was wondering, I'm looking at these atlases, and I was wondering how these superficial white matter bundles are named or classified, or is there a classification, or is it always named by the gyrus and then its neighboring gyrus, or do we know any of that from tractography? Do we know any of that from before tractography, tracing studies and things like that? So just naming and classification of these things. Or is it a sheet? Are they named based on the cortex it's under or something?
Yes, I think there are some bundles that have names. Yes, the vertical, occipital, fascicle, the Aslan tract. I don't know the name, I confess that I have stuck with that. But yes, there are some bundles that have names, but in general, we don't have a classification, and this is a problem. Also, even for the different lengths and the categories in the papers, for example, some papers talk about intragyral, intergyral bundles and medium range, short fibers, U fibers. So yes, I think we need a definition of the bundles. Yes.
And do you happen to know how so I see 500 bundles, 198. What are those named, or are they just named bundle 1, 2, 3, 4, or 5?
For this case, these are named in function of the connected gyri.
Okay.
And a number just based on the location, relative location of the bundle.
It gives the definitions unit a lot of work to do here.
Yes, I think absolutely. Yeah.
Thank you. Lauren, you can go ahead.
Thanks. Yeah, first I want to say hi, Pamela. It's nice to see you. And I thought this was an awesome talk. And I learned a bit about the different cells, and I hadn't known about the superficial white matter. So I was really happy I got to come. Yeah, thanks for including the picture of our atlas there. Yeah, and Kurt, our atlas, like hers, it's like numbers and just which gyri do they connect. But I got a little distracted by this conversation, but I also wanted to say, I'd love to talk more about it in the future. And Pamela, if you're interested, you're probably busy, but if you're interested in ever chatting more about the nomenclature of it, we're all very happy to chat with you. Or nomenclature, however you even say that word. Everybody says that word different, which is kind of ironic. Okay, so to continue on, I also wanted to just mention that the resolution of the diffusion MRI plays a role in what you can trace. I don't know if you had a slide about that or not, but that we found that with higher resolution submillimeter, you can trace some bundles that are down to like 10 millimeters in length that are closer to the cortex. So that just adds another level of complexity, because like these bundles here, we could see probably even in a lot of like clinical acquisitions, because you were saying there's a lot of these are medium range, but as we get shorter and closer to the cortex, there's even more challenges, and how do we depict those? So I guess I just wanted to mention that, and also see what your thoughts were on that kind of, how close to the cortex do we need to get? How short of a bundle are we actually hoping to ever be able to study? Or what are your thoughts on that whole sort of resolution and size question?
It's a good question. I think there is a difference between what we can do with clinical data and what we can do with more high quality data. I think maybe we need to differentiate the methods, because I think for clinical data we need simple and effective method to analyze the superficial white matter. Maybe I don't know, a few set of bundles and with a good segmentation, and that is but it's very interesting to go deep in the identification and understanding of these bundles with as we mentioned, the high quality database, and I would be really interested in also in see this data. It's very interesting. But I don't, I don't know too much this data, but I think the works that make histology and image, ex vivo image acquisition at the same time are very valuable to understand this kind of connections. Yeah.
Thank you. Thank you. Really nice to see you. I loved the talk.
Thank you. Nice to see you.
Thank you. There is a raised hand from Matthew. You can go ahead.
Hi, really cool talk. I was wondering, has there been much work trying to, I guess, recreate using tractography any sort of histology or tract tracing features of superficial white matter? So like maybe some histology studies that have shown interconnected areas within the same region. Have people tried to, I guess, recreate those with tractography? And has there been much success with that?
Sorry, I did not understand the question.
So areas that are connected, shown in histology or tract tracing studies, has there been much success trying to route, like, I guess, use tractography to make the same connections or the same superficial white matter bundles?
I think in this case with tracing, it has been done only for macaque or other animal models. I confess I have not went in deep in this kind of analysis. They can trace, but the problem is that they can trace a particular only a few fibers. But maybe somebody here can give a better answer.
Maybe Carlo wants to comment on your macaque data and some of the challenges we've used your data for. I think there is some evidence out there, but don't want to put you on the spot, Carlo, if you're there. I see your name.
It would be a fake.
I'm the Carlo who is put on the spot.
Yes.
Okay.
Thank you.
But I mean, my general comments is this one that unfortunately in the human brain, right, you cannot really do any invasive study that are necessary to look at the sparse connections. So everything we have in our mind about superficial white matter fibers, U fibers, longer range fibers, is something that is mostly in our construction in a way, and based also on some animal models. So that is the only thing I can comment upon, that unfortunately, again, for the human brain, I don't think that we have really a gold standard to go by. And that is the problem. Again, there are some, tracing studies done in monkeys and other species that could be informative. And keep in mind, again, when I studied anatomy and medical school, my professor, was telling me about some, pathways that we actually also in the tractography, MRI tractography realm, we started using this concept of bundles. In reality, again, we know that connections in the central nervous system do not go by bundles; they are sparse. And so the sparsity thing is the other aspect. But they say, again, some of the dentorubothalamic tract that actually people now say they are targeting for the, Margifas therapy of essential tremor, was indeed only demonstrated in cats, not in humans. So again, we have a very limited knowledge of what's going on in humans. So this is my 2 cents. But again, I'm not an expert. And the other aspect is that most of the anatomists now are pathologists. So they do microanatomy. They do not do big system anatomy, right? So again, beside what we have historically with Dejeran and so on, we really do not have very recent data in terms of gold standards beside what we do with MRI. So these are my comments. But again, congratulations again for your, and your references are indeed very important because actually, yeah, we need to see when you state something, right? It's very important to see on what basis this was stated, which type of studies were supporting that, were animal studies, human studies, and so on. But anyway, very interesting topic.
Thank you, Carlo.
Thank you very much. Is there any other question from the room? I see. Okay, you can go ahead.
Hey, Pamela. Thank you for this
Hi.
very much. Hi. So my question is related to your page 6. Can you go back a little?
Yes.
Yeah, I think on that slide you showed. Oh, here it's about this intracortical horizontal fibers. Yes. So my question is, from your experience, have you seen such kind of fibers that can be reconstructed using tractography? Have seen this kind of fiber before?
With the data that I have available, no.
Okay.
No. Maybe with more high quality data, I don't know.
You're talking about C here, is this correct? Fan?
This intracortical horizontal fibers, like inside the gray matter cortex.
Oh, A.
Yeah.
A. Okay.
Yeah. Yeah.
It seems there is a study with for the cerebellum that has used a very high field in postmortem data that they had tracked some cortical fibers, but
yeah, I mean, the cerebellum has, yes, intracortical horizontal fibers, which are the parallel fibers. So the cerebellum is very unique in terms of cerebral cortex. There are, horizontal dendrites, branches. As far as I know, there are no axons, intracortical axons that go parallel to the surface of the cortex. But yes, the cerebellum is a completely different architecture. So if you're referring to the cerebellum, yes, that is a dominant component. These are the so-called parallel fibers in the cerebellum. Yes.
Great. So thank you very much for the questions. I think there is no one else with question. It's 9:57, so I think we can conclude here. Thank you very much, Pamela, for your great presentation and for all the questions.
Thank you for your invitation. I confess that this was an intensive course of superficial white matter.
Well, thank you for all of the updated reference. Very appreciated. And we will not meet next month for Tract-Anat Club, so don't expect an email. We should get an invitation for January, and I keep inviting more people. Thank you everyone for coming, and have a great rest of the week.
Bye. Merci beaucoup. Thanks, Pamela.
Thank you. Bye.
Thank you. Bye.
Thank you.
Thank you again.